My name is Simon Roux. I'm with the Metagenome program and the viral genomics group at JGI. And today we'll talk about, as I was saying, this IMG VR database and tell you a little more about the background, why we do this and how we do it basically, and why it could be helpful in your research. I like to start with this, and typically, I'm telling people, hey, that's what usually comes to your mind when I mention viruses. Of course, right now, I don't even have to put the name of this virus here, just a picture. But these are not the viruses I care about, at least not now and not for this presentation. And again, for the very general background, the reason why is we as humans are a very tiny portion of life on Earth. Most of life on Earth is microbial. And that's true in the environment, and that's actually true inside human bodies. And linked to this is, since most of life is microbial, most viruses infect microbes. And again, human viruses are, of course, strategically important, but just a very tiny fraction of virus diversity on Earth. If you want a visual, not even representation, but an actual view of viruses, this is what we typically show. So what this picture is one drop of seawater concentrated and stained with cybergreen. Cybergreen with stained DNA, you will have microbes coming up as big blobs like this, and that's an interesting eukaryotic microbes here you see elongated. All the small dots behind, all these things here, which are regular small dots, these are viruses. So basically, this drop of seawater is just like microbes floating into a sea of viruses everywhere. If you like numbers, basically we have billions of viruses in every milliliter of or gram of everything we have looked at so far on Earth. So that's just my typical introduction of like virus diversity is gigantic and we have viruses everywhere. Of course, viruses are just not here, they are doing stuff. We need to understand their impact on microbes, on the kind of different areas or related but different impacts, I should say. Viruses will kill some microbes, which will reshuffle microbial communities and populations. Viruses will do horizontal gene transfer, so they will help take one gene from microbe A and transfer it to microbe B. And they will also reprogram the host cell during infection. When a virus comes into a host cell and transform it into a virus factory, it just fundamentally alters the way this microbial cell behave in terms of energy consumption and nutrient cycles and so on and so forth. So that's why viruses matter, basically. How do we study viruses in the environment? Well, it's not easy, actually, or at least it wasn't easy until metagenomics. For people familiar with microbial ecology, you may have, of course, be very familiar, or you may be very familiar with 16S and 18S amplicons and wonder why we didn't do that with viruses. The reason is like we don't have a universal marker genes like 16S or 18S or ITS. Of course, like every other microbe and microbe-related things, viruses are also difficult to cultivate. So both of these things, the lack of universal marker gene and challenges in cultivation, means we have still today a relatively poor coverage of virus diversity on Earth. And the way we typically look at this is by looking at it from the host perspective. So very briefly, this is a host tree. Again, I'm looking at viruses of bacteria in archaea here. These are different bacterial groups, different archaeal group. And that has to date a bit. It's like from 2016, but it's still relatively accurate. All the groups in red are the one for which we have at least one virus isolate. And so the flip side is everything in black has literally 0. And that's a lot of groups. That's basically what I want you to take from this picture. Again, that's all to tell you it's hard to look into virus diversity until metagenomics come along. Again, why metagenomics is perfectly appropriate is because we need a method that doesn't rely on cultivation and doesn't require any prior knowledge about the virus we want to study. And so that's what metagenomics provide. Again, very quick background. Metagenomics start with DNA, RNA extraction, and then shotgun sequencing and genome assembly, basically. So you start from your sample and you get pieces of genomes. If you really like viruses, you can add a pre-step, just before extracting the nucleic acids, you can add a step where you try to remove cells and just enrich for viruses. And that will, let you have more virus genomes at this step. It's recommended if you really want to look at your viruses. At the end of the day, what you have is a collection of genomes and sequences And what you want to do is find which one of them are viral sequences, and once you have identified these, then you can go on into like a number of different analyses. But I'll go through this kind of major step of like going from pieces of genomes to here are my virus genomes. And if you want to know more about the technical details about this, David Paez-Espino has published in 2017 a protocol to kind of perform this kind of virus sequence identification. There are also a number of other tools, but this one is really nice because it's really a step-by-step explanation of how and why you do this. Okay. First question you may have is like, I just told you that metagenomics is like solving everything and magical. You know, concretely, how does this translate? Is this really that good? And for that, I need to bring you a bit of historical context. Basically, viral genomes assembled from metagenomes have started in 2009, but it really was complicated except for very small viral genomes. And so it really picked up in 2016 with the first very large-scale study, which was led by JGI and David Paez-Espino, which was dubbed the HERS Virum. And that was going into IMG, looking at public metagenomes and collecting all the viral sequences we could find, or they could find actually. And that led to the establishment of the first version of IMG.VR, which is a database of viral sequences extracted at the time only from IMG. And to give you a sense of scale, so this is how it compared to isolates. The blue curve here is the complete total number of virus genomes in NCBI. So here I'm including phages, viruses of archaea, eukaryotic viruses, human viruses too. So that's your curve here, and it's 2016, 2017. We're on about 200,000 basically. And with the HERS Virum, it was already 125,000 virus genomes assembled. And by the time the IMG-VR first version came out, new data had been released. And so already here, you had more sequences of viruses in IMG-VR1 than you had isolates. And that, again, includes human viruses, which we comparatively study way more. But that was 2016. In 2018, there was a second version of IMG-VR, 750,000 sequences here. And then this V3 that I will talk about today is just like blowing the top of all this. We are now at 2.3 million sequences. And you can see the growth curve being extremely different between what we can do from metagenomes in this yellow color and what you can do from isolates in blue. So I'm not just like setting my stuff. Really, assembling viral genomes from metagenomes is becoming the primary way of exploring viral diversity at this point. Now, you may still have some question, and 2.3 million genomes seems super high. Like, is this really 2.3 good, really nice virus genomes? And the answer is like, not really, but still, it's still very good. Two things that are an issue when you do this kind of survey and when you try to explore viruses using this metagenome assembly. One is, if you do the same type of sample and you sequence the same type of samples over and over again, you may end up sequencing the same genome over and over again. So maybe it shouldn't be counted as different viruses. And the second is, for anyone who has looked into metagenome assembly, most people have been frustrated by small incomplete genome fragment. Basically, what we call a quote-unquote genome or sequence in my 2.3 million collection, some of them are very tiny pieces of much larger genomes. So how do we solve this? Well, instead of looking at all sequences, we can look at only the ones that are high quality, and we can make them non-redundant. And you have like the technical details at the bottom. What I call non-redundant is basically you can do pairwise genome comparison, and then you just cluster them when they looked almost identical. High quality, and I will come back to this a little later, but basically these are genomes that have been predicted to be more than 90% complete, which means basically these are not tiny pieces of much larger genomes. You can do it for NCBI, you can do this for IMG-VR, and you get this curve, which looks very similar to the previous one. The main difference is the y-axis here. You can see we are not in the millions range anymore. We have about 15,000 for NCBI, and we have 100,000 sequences for IMG-VR. So we still have the same ratio of like most sequences are obtained through metagenomes, even when you just focus on the non-redundant and high-quality genomes. Okay, so at least at this point, I should have convinced you that exploring virus diversity from metagenomes is one way to go, and the main way to go if you want to go high throughput and just do this very broad survey of every virus you can lay your hands on. So where do they all come from? As the meme is kind of hinting at, pretty much everywhere and anywhere. IMG VR3 includes sequences extracted from about 5,000 genomes and 15,000 metagenomes, all publicly available. If you look at a map, basically they come from all over the place, and this map is actually interactive and zoomable when you go to IMG VR, so I will show you this at the end. But if you look at more of the diversity in terms of environment, the majority of the sequences, 1.5 million, come from aquatic environment, and that's mostly a bias in the database. We'd have tons of metagenomes from aquatic environments, mostly marine and freshwater. Then you have sequences coming from host-associated. So what we call host-associated here is mostly human, as you would expect. So it's not human viruses per se, right? It's human microbiome viruses, so human guts mostly. And then sequences coming from soil and other terrestrial environments. And then what we have the engineered category here is a combination of wastewater and this kind of setup, or lab incubations. But basically, we have viruses from pretty much any environments you can name. Okay, now the interesting part, or at least the challenging part, kind of both. 2.3 million sequences. I don't think, like, at this point, it's not anymore something I can just dump into a file and share with a user on Google Drive and say, hey, have fun with this. Basically, this is too big to be usable just per se without, kind of advanced bioinformatics skills. So how do we make this like a resource that is useful for all users? Well, the first things we went after, or at least the main concept we walked around, is the fact that not all UVigs are equally useful. And again, I should have mentioned this. What I mean by UVigs here is my abbreviation for uncultivated viral genomes. So think of this as a viral genomes for which we don't have a cultured virus. But so not all these sequences are equally useful. Some of them are just short, like I was saying, and then some of them don't come from the right environment, or not in fact the right host for the studies that the user is trying to do. So what we went after is, for each sequence, we want to have a notion of quality, taxonomy, and host, and then we want to have an interface so that a user can very easily identify and extract the subset of these 2.3 million sequences that could be of interest for them. Okay, so I mentioned three things: quality, taxonomy, host prediction. Let's start with quality. So the quality of a genome in the virus world is something that we kind of outlined in a community paper or community-driven paper we led in 2019. And the idea is, at this point, what defines the quality of a virus genome is completeness. How much of the complete genome the sequence you have actually represents. No, it used to be a pain to calculate. It's becoming much better now, thanks to Stephen Neyfach, again from JGI, who created this tool called CheckV, which is the first and pretty much only tool which provides an automated estimation of completeness. And he also added, for good measure, detection of contamination. But here we are mostly using the completeness estimation. Basically, the idea is you can run your virus sequence through CheckV, and it will tell you, okay, your sequence represents 10% of the complete genome, 50%, or actually 100%, and that's a only a complete genome. We did that for IMG VR, again, the 2.3 million sequences. As you could expect, most of them came up as what we call your genome fragment, less than 50% complete. So basically, you have half of your genome in your sequence. So not that useful, maybe, depends on your application, but not the most useful ever. Then you have a small portion that is between 50 and 90%, and you have this 10% here, or nearly 10%, that are these high quality genomes. Again, that's the one I had in my second growth curve of the database. This one are more than 90% complete. So complete, like near complete genome at this point. So on the flip side, a majority of the sequences are low quality, but with the IMG VR v3, a user can very easily target and identify which of the sequences are the 10% that are high quality and 90% complete. That's the first step. Second step I mentioned, taxonomy. Taxonomy in the virus world is an interesting beast. mostly because it's extremely dynamic. These are just two examples of papers that came out actually in 2020. One completely changed the virus taxonomy and expanded it to what is now 15 taxonomic ranks. So we are now matching the cellular taxonomy, meaning we include realm, phylum, class, etc., which didn't exist before in virus taxonomy. And then in parallel to this, a team and a consortium actually led by Eugene Konin of NCBI proposed a complete reorganization of all the virus taxonomy, and which is now mostly based on genome-based features. So virus taxonomy historically has been based more on like biophysical properties, like the shape of the virus and how it infected the host and what it was actually doing to the host cell, which are interesting properties, but first, they are not necessarily good reflection of evolutionary history, and second, it's impossible to get these properties if you don't have the virus in culture. And the virus world and the official body of virus taxonomy, which is called the ICTV, accepted that we needed to shift to genome-based features so that we could meaningfully integrate UVIGs in the broader virus taxonomy. All that means, don't learn virus taxonomy right now because it's changing almost every month. But we still, we're able to work with people in ICTV to get the most up-to-date taxonomy to IMG.VR. That's a classification we made. So again, we classified our 2.3 million sequences. I don't want you to remember any of this, but basically what you can see here is a lot of blue. Blue here means what used to be called colorals, tailed phages. Basically, these are the most common type of phage or bacteriophages you can find in nature. So it's not a surprise to have, majority of blue here. What's more interesting in this plot is actually this part. So let me explain. This is only high quality genomes, so more than 90% complete, and still we couldn't place them in an existing taxon. So these are most likely completely new areas of the viral diversity that we have not yet explored or at least integrated meaningfully into taxonomy. So probably new families, maybe new classes, maybe new phyla, but a lot of taxonomic novelty being hidden in these large genomes, near complete genomes that we just can't classify at this point. Okay, I mentioned quality, taxonomy, now is the turn of host prediction. I think it's kind of intuitive for most people, but basically when you study viruses and virus genomes, one of the first questions you ask is, what is the host that is infected by virus A or B? If you don't know the hosts, there is only a limited number of things you can actually do or analysis you can actually perform on your viruses. So how do we do this? Again, I won't go into the technical details. I'm happy to discuss it offline or with questions, but basically we look at similarity between viruses, sequences, and host genomes and CRISPR spacers. The bottom line here is it doesn't work very well. And the more advanced, or at least the more detailed response is we do get prediction that we can trust mostly, but these predictions are only about a minority of our virus sequences. And in this case, in IMG v3, it's only 15% of our viruses. And that's because we just lack the corresponding or the relevant host genomes most of the times. So this is a bit more complex than this 1585. If you break it down again by completeness, you can actually see that for genome fragments that are less than 50% complete, so this kind of very short pieces of genomes, you have almost no host prediction. But for genome fragments that are 50 to 90% complete or more than 90% complete, this is much better. You get to like this 35%-ish range. And so it's not as bad as this 1585, although you can of course argue that that's a lot of gray basically here, a lot of sequences for which we have no clue what the host is. In terms of what are the hosts we find in IMG v3, again, we are looking at broadly any and every phage. And so when you look at predicted host, it's basically your usual suspects, the most common bacteria you can find, Proteobacteria, Bacteroides, Actinos, Cyanos, nothing completely crazy here. Okay, so not all UVIGs are equally useful. How do you know if a UVIG is useful for you? Well, quality, taxonomy, host prediction are the kind of three big features you need to know to sort through this massive database. And now we provide all three. So next step is, how do we present this to the user? And I've talked long enough, so I will actually try the live demo here. So IMG V3 has launched officially last Monday. And so far, we didn't have actually user response. So either no one has tried it or it's actually working. We'll see, I guess. This is your homepage. You just go to IMG and you click on VR and you get there. Here on the left, you have a few statistics, a nice drawings of viruses here. But so my point, since I've started to look at these 2.3 million sequences, is how do you get people to navigate such a big resource? So the first thing we have done is this thing here called Browse UVics. And basically at this point, it's for users who don't have a very specific question, but want to kind of navigate and go through the database and get a sense for what's in there. And so we let the user browse through taxonomy, ecosystem, predicted hosts, etc. So I had a screenshot for the geolocation before. I will just show you the actual map. But it looks something like this. So this is, again, map of the world, and individual pins. Blue pins are individual locations when you add one or more samples and one or more virus sequences. And then this colored circles are actually cases where you have too many locations next to each other to really resolve. So you can just then zoom, and this will resolve as you zoom in. And for instance, if you go to Greece randomly, you will see, for instance, here, okay, that's one location that's eventually got resolved as we zoomed in. And you can then click on this location. You have different metagenomes here, 3 different metagenomes. And then here are 8, 112, and 1 virus sequence on which you can then click. And that will take you to the actual virus sequence found in these metagenomes. That's the way you can just browse through this kind of geographic location aspect. Now that's, again, based on the metagenome of origin. I mentioned browse by host prediction. The way we do it is using these tree maps. So this tree maps is representing the phylogeny, or at least the taxonomy of your host. So you can start from the left. You have bacteria and archaea, then protobacteria, et cetera, et cetera. And the area associated with each taxon is proportional to the number of viruses predicted to infect this taxon. So you can just go through the tree map, or if you have a specific bug in mind, you can just search it on the top. Let's say you are JGI, so you like streptomyces. You can just click search and it will take you straight to streptomyces. Again, if you hover the tree map, you have a number on top. I should actually not point on my screen, but with the mouse. Number on top here, 305, is a number of virus sequences linked to this specific taxon. So you go down in the taxonomy, and this number will just go down as well. And so once you get to your clade, for instance, streptomyces, you really like this 286, you just click on it, and you will get a specific, a reduced table of just these 286 viral sequences. And again, we come back to the different features I highlighted in my talk. So basically, here in this table, every line is a virus genome. You get the genome or metagenome of origin. You get taxonomy here, some information about, is this a linear config? Is this integrated into the host genome? What is its length in base pair? What is its estimated completeness, et cetera, et cetera. Here is what is the actual environment. For instance, maybe here we just got like 286 viruses predicted to infect streptomyces. Maybe you want within this group the ones that have been found in soil. So you can just type soil here, click enter, and then you will get only the ones that are linked to metagenome, in soil ecosystem. And then you actually have some information about how these sequences were linked to streptomyces. So that's one way of kind of navigating through the database and narrowing down progressively to the subset of sequences you are really interested into. And once you have this list, you can export it in an Excel spreadsheet. You can get the sequences and do everything you usually do on IMG. So that's the kind of browse aspect. But maybe you came in and say, well, I don't need to browse anything. I know what I want. And that's where the search UVics will come into play. You have a different few different ways to you can search. If you have a sequence that's you're interested into, you have a virus sequence especially, and you want to know if there is a hit in the IMGPR database, you can just use this online blast here. But then maybe more interestingly, the search UVIG by UVIG attributes here. And that means actually being able to use multiple criteria to narrow down, again, these 2.3 million sequences to something that is actually interesting for you. So for instance, let's say you want something that is at least 80% complete, so from 80% to 100% complete. You want viruses that come from seawater, so marine viruses. You want them to be in the specific taxonomic group called Autography viridae, T7-like. And then you want them to infect gamma protobacteria, which the tree is so big that I just searched through it. And so at this point, again, I want this specific virus taxon. I want this host, gamma protobacteria. I want the viruses to come from a marine environment, and I want them to be 80 to 100% complete. And by doing this, this will let me just search and do this combined query, and it should give me back a very reduced table that only has the few viruses that I'm interested into. And in this case, it's only 12. So really just, again, 2.3 million and 12. And that's the whole point of this. At least that's the whole idea we had behind the interface we designed, is to be able to really just subset and narrow down this massive database. And with that, I will just have one final slide, which is just to say, these are also people who I need to thank for all the hard work in designing both the back end and the front end. And thank you for everyone.